Okay, welcome back to our next neurophysiology lecture. I'm recording this one right after the force of contraction one, so hopefully I can just upload these both at the same time for y'all. But during this lecture, we're going to start to build a little bit more of the foundations for neuro for us. We actually did do a mini kind of neuro intro at the beginning of our skeletal muscle unit, so a lot of stuff from that unit is going to come back today, and hopefully I'm able to give you more context. But I would recommend at some point going back to rewatch that lecture after this one, and I think it will make more sense after watching this one. But our nervous system is divided into our central nervous system and our peripheral nervous system. Our central nervous system is our brain and our spinal cord, and our peripheral nervous system is really everything else. And our peripheral nervous system has two divisions. It has an afferent division and an efferent division. The afferent division is our sensory, and our efferent is our somatic, motor, and autonomic divisions. So the terms afferent and efferent here, these are terms that you'll hear pretty commonly within neurophysiology, especially when you get into the nervous systems. These are important terms, and they are relative to the central nervous system. Now, what do I mean by that? Because they're part of the peripheral nervous system. So if we have the spinal cord, and this is part of the central nervous system, the spinal cord, of course, leads up to the brain. So we have an afferent division and an efferent division that can extend in or out of the spinal cord. So typically the sensory division of peripheral nervous system is what relays this information back to the spinal cord. So sensory information approaches the central nervous system. So this is why it's the afferent kind of arm of the peripheral nervous system. And then the spinal cord and brain can send out signals, and typically this exits the spinal cord, and this is going to be our somatic and autonomic branches. Somatic means motor system, right? So our ability to move. So somatic motor system. But these are exiting the central nervous system, which is why they're the efferent division. So I wanted to give you that little mnemonic because it helps me remember it. Sensory neurons approach the central nervous system, and motor neurons exit the central nervous system. So somatic and autonomic neurons. So afferents approach, efferents exit. Okay, and this is just a good diagram, just kind of it's kind of an overview diagram. I'm not expecting you to know exactly what this is showing with the actual sensory pathway, but I think this is a good indication of how your peripheral and central nervous system works together. So this is just if you were to see water, we would have sensory input from seeing water, and this would be visual sensory input. That sensory input is going to travel along neurons, travel down sensory neurons to approach the central nervous system. So here's our CNS. This one doesn't need to go through the spinal cord because the eyes are close to the brain. So this will just synapse directly into interneurons in the brain. So the brain, central nervous system, this is going to have interneurons. Interneurons. These interneurons receive information from the afferent, and then they send out efferent information, right? So this is the integrator. This is the master sort of getting information and then telling the body what to do with that information. So here, if we see a glass of water, we're going to sense that, send that information to our brain, we will process it. And then if we would like to drink it, right, then we have to send out a motor signal to the bicep, tell it to contract to pick up the glass of water. Right, so this is just a very generic neural pathway. You don't need to know this one. I just wanted to show you this to kind of show you a visual of the divisions between peripheral and central nervous system and sensory versus motor output, right? And this is kind of a nice tie back to our muscle unit previously, because the neuromuscular junction, right? We just learned all of that physiology on how a neuron is able to stimulate a muscle to cause it to contract. So our nervous system is one of two long-distance communication systems in our body. The first one is our nervous system, and the second one is our endocrine system. You'll actually explore your endocrine system more in A&P 2, but for now, we're doing our nervous system. So neurons, I have two pictures of neurons before on the slide here. One is myelinated, one is unmyelinated. We'll get to that a little bit later as we start to investigate neurons. But the way neurons communicate with each other is through electrical signals and chemical signals. So within a neuron, it conducts an electrical signal, right, known as an action potential, which is what you learned. And in chemical signals, this is going to be in between neurons. They have to communicate using neurotransmitters. So it uses diffusion of chemicals, which is a slightly slower process. This is analogous to when we had the neuron communicate with the skeletal muscle, right, in EC coupling. It was the chemical of acetylcholine that was communicating between the neuron and the skeletal muscle. So neurons are generally a fast form of communication, but the electrical signal is very fast, and the chemical signal is generally a little bit slower in between neurons. So I just wanted to remind you here that we have a term called membrane potential, and this is the difference in charge between a cell inside and outside of the cell, because the way a neuron is able to propagate an action potential, right, send this electrical signal down its cell, it is manipulating the membrane potential in order to do that. How does charge of the cell influence ion movement? So flux here is just movement, right? This is a fancy word for movement. And we know that things always move down their gradient, right? They move from a high end of the gradient to the low end of the gradient. This is what we talked about back in that intro neuro unit. But I want to give a little bit more context to how ions move. So I have two scenarios here for you, cell A and cell B, and I have the gradients for sodium shown here, so Na+. In both scenarios, we see the gradient is the same. 115 millimolar on the outside, 15 millimolar on the inside. So the magnitude of this gradient, direction and magnitude of this gradient is the same, right? So the size of it is the same. But then what's different between these two scenarios? Well, what's different is the membrane potential, right? So over here, the arrow that I've drawn so far, this is the concentration gradient, right? I've drawn it high to low, high on the outside, low on the inside. You might remember salty banana for sodium. But now we have to take into account the charge on the cell, because ions are charged particles. So in scenario A, we have the membrane potential as -70, and in scenario B, we have the membrane potential as -20. So my question here is, like, which one is cell A or cell B will have a greater flux of a greater movement of sodium into these cells? And because it's an ion, we don't just consider the concentration gradient, we also have to consider what's called the electrical gradient. To do that, we have sodium being a positively charged ion. We have the membrane potential, which is negatively charged. So we know how opposite charges interact, right? They attract. So in both cases, because we have a negatively charged membrane potential, the electrical gradient will also be pulling sodium into these cells, right? Because the negatively charged inside will attract the positively charged sodium on the outside. So we have something called the electrical gradient, which also can attract sodium in. Together, these are called the electrochemical gradients. The fact that there's just a normal concentration gradient, right, high to low, but there's also this electrical component that takes into account the charge of the ion and the charge of the cell membrane. Now, typically, electrochemical gradients do point in the same direction as the concentration gradient. That is until we get to what's called equilibrium. So you can imagine that this electrical gradient is, like, because it's another form of a gradient, it could either positively influence the overall gradient or it could, like, negatively influence the overall gradient. And let me give you an example, actually, before I go to the next slide. So if we had a cell here again, right, but now the membrane potential was +70, or maybe not that positive, let's just say like +30, and we still had sodium high outside. Sorry, it's just a plus charge. All right, we have sodium high outside, low inside. So the concentration gradient for sodium, just looking at just the concentrations, would be high to low. But now the electrical gradient the electrical gradient, we have to consider the charge of this. It's a positive charge. Now, this positive charge is going to repel sodium. So this positive charge is actually going to have influence outside of sodium. So it's going to try to push sodium out. Now, whether or not this electrical gradient is actually able to overcome that concentration gradient, that's another story. And in mammalian physiology, we actually really don't see any examples, at least not in basic anatomy and physiology classes. We don't see examples of the electrical gradient overpowering the concentration gradient of an ion. So the electrical chemical gradient will always face typically in the same direction as just the concentration gradient. There are some niche cases in physiology where this does happen. There are some examples in plants where electrical gradients can actually overpower concentration gradients. But I want to just show this to you because a lot of what we do with ion movement, we really have to understand concentration gradients and electrical gradients. So in this case, the concentration gradient would overpower the electrical gradient, so you would still have like a net movement in for electrochemical gradient. Electrochemical gradient. And if you're wondering how I know that, I haven't given you all of the context yet, right? And that's what this slide is for. So I have to tell you about something called equilibrium potentials. I'm going to show you an equation. Well, it's on the screen. I don't expect for you to know this equation. I'm not going to ask you to solve it, but conceptually, I would like you to know what it does. And I'm going to talk to you about the equation relative to just two ions, although this equation can be solved for any ion that's in an environment. But what this equation is called the Nernst equation. Nernst equation. And when we solve for it, we solve for something called an equilibrium potential. So an equilibrium potential, this is like a fundamental concept within neurophysiology, and it's a quite difficult concept to grasp. But let's kind of break it down. So for potentials, we know this is just the term for membrane potential, right? Membrane potential is charge of the cell. Equilibrium now, this means no net movement. No net movement. So what is equilibrium potential? It is the charge of the cell for which there's no net movement of the ion. So equilibrium potential is the charge of the cell for which there's no net movement of a given ion. And this is different from ion to ion. So sodium and potassium each have their own unique equilibrium potentials. So the equilibrium potential, this is the subscript for equilibrium potential, for sodium is equal to positive 61.5 millivolts. And really just positive 61. And the equilibrium potential for potassium here is equal to minus 90 millivolts. So let's kind of think about like what these numbers mean. So if a cell is at, let's say, minus 80 millivolts, let's look at potassium first. So potassium's gradient is typically high inside, low outside. Let's say the membrane potential is currently minus 80. So just based on concentration gradient, we would have potassium moving out of the cell, right? So it's moving out of the cell just based on its concentration gradient. The membrane potential, though, is minus 80. So if we were to write an arrow for the electrical gradient here, we would be actually attracting potassium back in, right? Electrical gradient. And the magnitude, the size and magnitude of this is minus 80 millivolts. So this is a minus 80 millivolt electrical gradient. It's pulling potassium in this case because we have opposite charges, right? Potassium's positive, the membrane potential is negative, so opposites attract. This would be an attractive force into the cell. So if we have the CG and the EG here, how do we know which direction potassium is actually going? Well, I had told you before that, the overall electrochemical gradient is always aligned with the direction of the concentration gradient, but let me show you why. So what the Nernst equation is actually doing, yes, it's solving for equilibrium potential, but it's also doing another key aspect here. The Nernst equation is also a unit conversion of concentrations to millivolts for membrane potential. So we see that actually part of the Nernst equation, we have to plug in the concentration of said ion outside of the cell and the concentration of said ion inside of the cell. And our final result is a number in millivolts. So it's a membrane potential, which is what that is. It's just voltage of the membrane over there. So the Nernst equation is calculating the equilibrium potential, the membrane potential at which there's no net movement, by using the concentration gradient. So what this equation is really saying conceptually, it was like this charge on the cell for this ion is where the ion will no longer move. Right there, the rate of it moving it in and the rate of it moving out are equal and opposite, so there is no net movement. So for potassium here, when solving the Nernst equation, you don't need to solve it. I already solved it for you. It's about minus 90 millivolts. This is the unit conversion of the CG, right, from millimolar into millivolts. So the magnitude of this CG arrow is actually 90 millivolts. It's technically a minus sign on it, but it's facing out, right, facing out. Whereas the minus 80 is facing in. So we could just kind of visually look at these two numbers and compare them. Well, we have the force of this concentration gradient pointing out, and the magnitude of that force is 90 millivolts. We have an attractive force pointing in, and the magnitude of that is 80 millivolts. 90 is bigger than 80, so which one overpowers which? The CG overpowers the EG. So we would have a net ECG here that's pointing out. The net ECG here, the magnitude on it, it would be pretty small. It would only be about 10 millivolts, right? Because that's the difference between these two. And now potassium is moving out of the cell through, of course, there would need to be channels there, right? But through that net force of the electrochemical gradient. Let's look at the same thing again for potassium, but now what if the membrane potential is minus 90? Again, we have potassium high inside of the cell, potassium low outside of the cell. The concentration gradient is always just whatever is high to low. And now we know that the magnitude of the concentration gradient is minus 90 millivolts. Again, this is pointing out of the cell. And now we have this minus 90 on the cell membrane. This is an attractive force. So the electrical gradient is an attractive force pointing in. And the magnitude of this is 90. I'm putting the minus sign on here. The minus sign doesn't fully matter here. It's just kind of the size of the number, right? When I say magnitude, I mean the size of the number. So if we have something that's 90 pointing out and 90 pointing in, you can see that these are equal and opposite, right? So this is equal and opposite. So if these are equal and opposite forces to each other, this will result in no net movement for potassium ion. And you can imagine if the electrical gradient now, you can imagine if this became one higher, if this became like negative 91, it would start to reverse the gradient in the other direction. So potassium would actually be pulled in where the electrical gradient is overpowering the concentration gradient. Again, like I said, this typically doesn't happen in mammalian physiology. Okay, so for neurons, we need to go over sort of the basic anatomy of a neuron. And I have these terms here on the side that we're going to label. So for both of these, we have a multipolar neuron on the left and a pseudo-unipolar on the right. These would be an example of like a peripheral nervous system neuron and then like a central nervous system neuron, because the central nervous system is all like interneurons, and these interneurons are like relaying information from one side and sending it to the other. Whereas the multipolar neurons are going to be the ones that are like the sensory or motor ones. So we have these terms here, and they're going to be found on both neurons, so we'll label them together. The sensory portion of neurons is called the dendrites. So that's going to be over here. These are the dendrites. And when I say sensory portion, I don't mean like they're part of the sensory system, but that they are communicating from whatever's behind them. So that could be another neuron, or it could be the like open world. So the dendrites are the sensory portion. The cell body is going to be here with the nucleus. And over here, cell body. The trigger zone is the very beginning of the axon. So trigger zone. Trigger zone is the very beginning of the axon. If you look at different resources, or if you've happened to study neurophysiology before, you may have heard of this region referred to as the axon hillock, which is also true. It's just the very beginning of the axon. And the axon is the long extension from the cell body. Well, I guess in the case of the multipolar neuron, right, it's from the cell body. But the axon is just the really long extension of the cell. The axon terminal is going to be the end portion here. So I wouldn't be able to expect you to do this for like an interneuron, because it's a little bit hard to tell what the dendrites versus the axon terminal are. That's why I kind of rotated this picture of the pseudo-unipolar neuron so that it looked in line with the multipolar neuron, so we could see the difference between the dendrites and the axon terminal. Functionally, they do different things, but anatomically on the ones on the right, they're a little bit hard to tell the difference. Okay, so we have this slide here to define some of these terms, or I guess just to remind ourselves of the definition of some of these terms. So we have it over here, right? We have like if a cell was at rest, and then it depolarized, and then it repolarized, and then it hyperpolarized. But just so we have these definitions down in our notes, a depolarization is becoming less negative. Any movement of membrane potential that's less negative is referred to as a depolarization. Repolarization is more negative. And then hyperpolarization is more negative than resting membrane potential. And resting membrane potential in mammalian cells is typically minus 70 millivolts. So if anything is below that, we would say it's hyperpolarized. And then if it's moving up or down, that would be a depolarization or a repolarization, respectively. So let's talk about like what is happening at cells when they're at rest. So like I said on the last slide, the resting membrane potential for cells is typically around minus 70. So if we just have a cell that is down here at rest, what we need to think about is like, well, like what is contributing to this rest? So we have, when the cell's at rest, it could be stimulated, right? And if it gets stimulated past a certain threshold, then it will fire an action potential. So above that threshold is the action potential, and below that threshold is something called a little bit different, but we'll talk about that in a second. So like we need to really investigate like what is happening to these cells at rest and what's helping maintain resting membrane potential. So within cells, we actually did talk about this a bit on the last sort of intro to neuro lecture, but now we have more context to it. So on a given cell, we have the typical ion concentrations, which are shown on the screen. And then we have certain proteins here. Let me label some of these proteins for us. We have the sodium potassium pump. That's a plus. And then we have potassium leak channels. And we have sodium leak channels. Sodium leak channels. channels. So whenever we're deciding on, which ion is influencing membrane potential at any given time, we have to consider the permeability for that ion. And for permeability, we have to consider how many channels for that ion are open or closed. And they could be present or not present on the cell membrane. So for potassium, let's look at like resting membrane potential a little bit more. Because what we want to decide is, for a cell at rest, do we think proportionally there are more potassium ions or sodium ions for leak channels? Because I told you before that there are leak channels present on all of these cells. So if resting membrane potential is minus 70, I have to introduce a sort of another key foundational concept within neurophysiology that I'm going to write this down for us. So at any point in time, the membrane potential of a cell is going to be closest to or approaching the equilibrium potential of the most permeable ion. So this is a really foundational concept within neurophysiology. So at any point in time, whether the cell's at rest, whether the cell is depolarizing, repolarizing, hyperpolarizing, the cell is always going to be closest to or approaching, like on its way to the equilibrium potential of the most permeable ion. So the equilibrium potential of sodium is positive 61.5, of potassium it's negative 90, negative 91. We can kind of use them interchangeably because it's like technically in between. So whichever one you want here. So negative 90. So if we have a resting cell is at minus 70, we see that it's a lot closer to the equilibrium potential of potassium at minus 90. It's a lot further from the equilibrium potential of sodium at positive 61. So we can reason that if this is true, which it is, this is true for all neurons, all cells at all times, if a cell's resting membrane potential is minus 70, that means that it must be most permeable to potassium while it's at rest. So the main channel that's contributing to resting membrane potential is potassium leak channels. Potassium leak channels. So if you messed with the proportion of potassium to sodium leak channels, that would adjust resting membrane potential. You could either raise it or lower it if you messed with the concentration of each of those channels. Okay, so now we're going to get into kind of giving you a little bit more information about how a neuron generates an action potential. And every action potential actually has to start with what's called a graded potential. So a graded potential is what occurs in the dendrites of the cell, and the strength of the graded potential is dependent on the stimulus. So in other words, it exists on a grade, right? The stimulus could be really strong, that could cause a big graded potential. The stimulus could be really weak, and that causes a weak graded potential. These are typically started by mechano or ligand-gated channels, but it is not limited to those. And a really good analogy for a graded potential is that, like if you drop like a stone in water, right, the little ripples here get smaller the further away from the like origin of the drop. And that's similarly what's happening, but with the electrical current of a graded potential within a neuron. Wherever that ion enters, it's going to cause kind of the biggest increases in membrane potential, but it's going to get smaller and smaller the further away you go. And I have a visual for that here. We have like neuron 1, and then we have neuron 2. Neuron 2 is shown in like way bigger, right? It's showing like zoomed in on that. And we have neuron 1 firing an action potential, releasing that stimulus onto neuron 2. And we see this influx of sodium. But what we see here is that we have like this wave-like pattern, right? Where it gets smaller and smaller the further away from the origin of the stimulus that we get. So what happens is this current, this electrical current, it just kind of naturally dissipates for a graded potential. You don't need to know why it naturally dissipates, although you might be wondering why, so I can give you some, reasons why. This is just FYI in case you're curious. So if the sodium is traveling in all directions throughout the cell, you can imagine that it's going to encounter a lot of resistance, right? Like just like waves flowing through water, like there is an inherent resistance to electrical current moving through this fluid. And the other thing too is that there are channels, right, on the cell membrane, and some of that sodium that comes in might just end up exiting that channel somewhere else down the line. And if it does, right, that would help dissipate the strength of the signal. The other thing too is that there are charged particles within the cell, right? There are negatively charged like proteins, and some of those proteins might start to have ionic interactions with a positively charged sodium that's trying to move through the cell membrane, or near the cell membrane through the cell. So there's a lot of different reasons why this charge dissipates, but the point is a graded potential, it does dissipate. It decays with distance. Right? The further away from the origin of the signal, the weaker the stimulus is. And what we're going to kind of assume in most cases is that the graded potential is strong enough to result in an action potential. So graded potential must be strong enough to generate action potential. In most cases it is, but there are some scenarios where it's not strong enough to generate an action potential. So when I say here that the graded potential has to be strong enough, I mean it has to reach threshold. Right? And threshold is the membrane potential required to open voltage-gated sodium channels. So now we're going to go to action potentials, but just to kind of recap, if we were to divide this picture in half, here is where we have the graded potential happening. And then down the axon is where we have the action potential. This neuron happens to be myelinated, but not all neurons are myelinated, so it just kind of depends. But here's some key aspects of the action potential, especially relative to the graded, which we just talked about. Action potentials are all or none. Once they start, they do not stop until it reaches the end of the neuron. And they do not decay with distance. So no matter how big the neuron is, once the action potential starts, it will just propagate and fire down the entire length of the axon. It occurs in the axon, and it starts at the trigger zone, right? So that trigger zone would be right there at the very beginning. Trigger zone. And the action potential cannot vary in strength, right? Like the graded potential could vary in strength, but the action potential cannot. It always has a set strength, a set amplitude, and it fires at that amplitude every single time. And it's produced typically by voltage-gated channels, right? Voltage-gated sodium and potassium channels are what will help propagate the action potential down the length of the axon. So for action potentials, this is actually going to be identical to what we learned back in our intro to skeletal muscles lecture. So we have two neurons here. I'm going to kind of redraw and talk about it again. So this will be a good review for you to study action potentials. So let's imagine we had like some sort of stimulus here. So the cell starts off at rest, right? And this is due to potassium leak channels. But once it gets the stimulus, it's going to start to increase its membrane potential. And this is going to be our graded potential. Graded potential. Graded potentials, especially if they're positive here, they're typically started by sodium, or ligand-gated sodium channels is what causes the graded potential. And that graded potential is going to happen here until we get to the trigger zone. Once we hit the trigger zone, we're now going to switch into our action potential. So I'll change colors for this. And we're going to have this fast depolarization. And this is due to voltage-gated sodium channels. So sodium is going to start to enter the axon at all points where we have these open channels. Eventually, those are going to close, right? You might remember that I told you up here we have two events happen. We have the voltage-gated sodium closes, and I'll change colors, maybe green. Our second event is voltage-gated potassium opens. Once the voltage-gated potassium opens, it will repolarize, hyperpolarize, and then return to rest. And again, this repolarization here is due to voltage-gated potassium channels. It's going to repolarize it. It's going to overshoot resting membrane potential, and this whole portion here is going to be called hyperpolarization. Oh. And now you actually have a little bit more context behind this hyperpolarization, right? You see that potassium. You see that potassium is overshooting the resting membrane potential and going down to minus 90. And you know the significance of minus 90 now for potassium, that this is the membrane potential at which it no longer moves, or right, that the net movement in is equal and opposite to the net movement out. So that the overall, there's no net movement for potassium. And so we can see that if we just let these channels stay open, the cell will eventually just reach the equilibrium potential for the most permeable ion at that point, right? And then for sodium on the other side, sodium's equilibrium potential is actually up here. Let me switch back to blue. It's actually up here, right at like positive 61.5 millivolts. But if we were to leave those voltage-gated sodium channels open for longer, these cells would have just reached 61.5, right? So this goes back to the idea that at any point in time, the cell's membrane potential is going to be closest or approaching the equilibrium potential of the most permeable ion. So in blue, the most permeable ion is sodium. In green, the most permeable ion is potassium, for the action potential portion of this, right? Okay, so that was hopefully a good review for you for action potentials. I have another graph here for another neuron, and I kind of just put it here to see if you would like to practice drawing a second one. So could you talk through the mechanism of, like, okay, now that the action potential reaches here, how does calcium enter the axon terminal? How does that cause exocytosis, right, of all of these? My pen was acting a little slow. How does that cause exocytosis of neurotransmitter? How does that bind to the ligand-gated ion channels on the next one, and then eventually cause an action potential to propagate down the next neuron? So I would say try to use the second half of the slide to review for yourself. Here we have the events at the end of the axon terminal. These are actually also just review slides for you because we covered this in the neuromuscular junction portion. So again, this is a good place to remind yourself, can you go through the flow diagram of action potential propagates down the presynaptic neuron? I'll help you get started here. This is going to open voltage-gated calcium channels, and then what happens to get to here? There's a lot of blank space. It might not be that big of a flow diagram, but this is just kind of the start and the end. And then once you get to that neurotransmitter being released into the synapse, what are your steps in order to get to, we now have an action potential propagate down the next neuron? So these are some slides to help you review like neuron to neuron communication and to remind you of the mechanism of neurotransmitter release. Very, very similar to the neuromuscular junction, but we don't have a neuron to a muscle this time. We have a neuron to a neuron. So it's a little different anatomically. Okay, I want to talk about another concept called refractory periods for neurons. For a refractory period, by definition, refractory period is the time when another action potential cannot happen. So with action potentials, they, like I was saying kind of at the beginning of action potentials, they cannot stack, right? They can't increase their amplitude. So if we're like currently going through, right, if this is like the duration of an action potential, if we're like currently in an action potential, we can't have another, right? Because like you can only have one at a time. So because the channels are opening and closing and they have to finish that action potential before they can fire another one. So if that's the case, during the action potential, this is just what's called a refractory period. Refractory period. Now, refractory period is just the time at which another action potential can't occur because one is already happening. There's also something called temporal summation and spatial summation for neurons. So I'm going to talk about that over the next couple slides. And it is similar in concept to temporal and spatial summation in skeletal muscles, but here we're not dealing with force generation, we're dealing with action potential frequency. So before how we said, okay, this cell's at rest, and then it got an excitatory signal and it just approached threshold. It went up to threshold and then yay, action potential, right? So that's what we did before. Well, I actually kind of simplified things a bit when I said it just gets the signal and increases all the way to threshold. That doesn't actually always happen. So if we have like neuron 1 and neuron, let me relabel this. We have actually neuron 1 up here and then neuron 2 right here, and we're like recording the membrane potential of neuron 2. It starts at rest, and then we see it's actually getting repeated signals from the previous neuron. So these repeated signals end up stacking on each other. So this is actually the graded potential portion of it. So instead of it just looking like this, where it goes straight up, what it's actually doing is like it's increasing a little bit, a little bit, a little bit, a little bit, and hopefully it's enough to reach threshold. And when it's increasing a little bit and a little bit, all of this is actually the graded potential. And you can see now that I simplified it earlier by saying, oh, it got a signal, just shot straight up to threshold. But in reality, the previous neuron needs to stimulate it multiple times in order to get it to threshold. And the reason why it needs to stimulate it multiple times is because it can decay, right? So if it was, let's say if it was at rest and it started to get some signals, maybe that excitatory signal stopped halfway through and then it just decayed back down to rest. If that's the case, we actually never reach threshold here. Never reached threshold. So we wouldn't have an action potential in that case because the signal decayed before it got strong enough by the time it reached the axon hillock, right? Okay, so this is this idea of temporal summation is just the previous neuron stimulating the next neuron frequently over time, right? You add them all together over time. Spatial summation is really similar, where you just have, if we have like the main neuron there, we have 1, 2, 3 neurons talking to neuron 4. And if we had the graph, right, threshold, minus 70, it's at rest, it would be getting 3 excitatory signals at the same time. So it'd like jump up even faster. Right, so that graded potential would be even faster if there were multiple neurons there contributing to the graded potential. So because the graded potential is summating across space, right, 1, 2, 3 neurons before, we call this spatial summation. Very similar to skeletal muscles. Whenever a neuron is firing an action potential, both temporal and spatial summation are playing a role. Right? Each individual neuron is giving a certain frequency, and if you have multiple neurons, then that adds to the layer of spatial summation. Both of these are techniques that neurons use to reach threshold to fire an action potential. Okay, I think this is the last slide. My voice is like really starting to go out now. but hopefully we can get through this. Okay, so this is kind of another example of spatial summation. We have 1, 2, 3 previous neurons interacting with the fourth neuron. But notice something about neuron number 3. This is an inhibitory neuron. So when it releases its neurotransmitter, it releases an inhibitory neurotransmitter that will hyperpolarize the next cell. So we actually have this battle for neuron 4 between 2 excitatory neurons, 1 inhibitory neuron. They are going to spatially summate and maybe or maybe not reach threshold. So the way this would look on a graph, right, is we're here at rest, and maybe we have a few excitatory signals, and then we have some inhibitory, and then maybe excitatory and inhibitory, excitatory, inhibitory. So maybe we have a few excitatory signals, and then we have a few inhibitory. After the inhibitory, maybe we have some excitatory, and then inhibitory back down, excitatory, inhibitory, and then excitatory, maybe up to threshold, right? Then we can cross threshold. What a graded potential actually is the summation between all excitatory and inhibitory signals. And these excitatory and inhibitory signals have specific names. So the excitatory portion is called let me give myself more room here. It's called an excitatory post-synaptic potential, abbreviated as EPSP, excitatory postsynaptic potential. And the inhibitory portions of this, where we're going downwards, are called inhibitory. Inhibitory postsynaptic potential, also known as an IPSP. So what a graded potential is actually all EPSPs plus IPSPs. It's the summation of all EPSP and IPSPs. And these could either summate to be greater than threshold, or they could summate to be lower than threshold. Again, this is why we call them graded potentials. It exists on a grade. There could be high ones, there could be low ones. The low ones would not generate an action potential. The high ones would generate an action potential. Okay, good job with neurophysiology here.